Hello everyone, thank you for the kind introduction. So my name is Shahar Harel, I'm the head of AI at Curis, a computer scientist and machine learning researcher background. My research focus was about deep generative models for generation of novel chemical compounds back in 2016-17, before generative models became the buzz in machine learning. And today I would not like to talk about buzz. I would like to talk really about how artificial intelligence connects in vitro, state-of-the-art in vitro work and state-of-the-art in silico work in order to generate a better MPS screening process and eventually a better drug safety prediction platform. So I will start by showing you how Curis uses machine learning to fuse those two paradigms, those two huge modalities of in silico, of in vitro, and following that, I will briefly discuss some of the main challenges that we are tackling today using machine learning to solve some of the most difficult challenges in the field. So to start showing you what's the focus of our platform, in the core of our technology is a drug safety prediction platform. It has three main phases. The first phase, where we generate data on the MPS, we make interaction between drugs in various dosages with the system and generate data. We then train the machine learning model on this self-generated data, and eventually, based on the model that was pre-trained and the new drug candidate, we can predict for this new drug what will be its toxicity profile. Let's dive a little bit into this process to understand it better. So as input to the process, we have thousands of drugs already labeled to some aspect of their safety or toxicity. You can think about drugs that have been through clinical trials, went to the market, and are now labeled by the FDA for their level of DILI, but this is just one example. Everything that failed in clinical trial or passed, we know the reasons. So we have an initial population that we have knowledge about regarding their safety profile to start this. We are in process of scanning each one of those drugs on our MPS platform. What's special about our MPS platform? So I will not get deep into the chip itself. Our VP Biology is also here. You are welcome to talk with him afterwards, but I want to concentrate on how we are using machine learning on top of this. So we are screening each one of those drugs on our platform, meaning we are generating the interaction between the drug in various dosages and the MPS. Remember, this screening process from our view has two goals. One, to best reliably mimic the physiological aspects of the human body. So this is, I assume every MPS attempts to do that. But no less important, we want to generate a data set that will have the predictive signals that the machine learning will be able to do something with it afterwards. So we are balancing actually between those two. And one important side beside the biological part of the chip is the data collection part. So we have multi-data collection modalities. One time series of microscopy images done with multiple imaging methods. A multivariate time series of biomarkers. You can think about time series of glucose, lactate, albumin, and so forth. And the chemistry, from the molecule that went into the plate, the drug itself, and what it's been through the body, for example, by reacting with liver enzymes. So those data modalities, and this list keeps growing, but those three are currently the main data modalities that we are creating, that we are measuring during the screening process. We end up with a huge data set. You can think about those thousands of drugs in rows, and in the columns, all the features that we have created on the MPS platform, and the features that we have computed on top of the chemical structure in silico. I will go deeper into how do we do it, but for now, what's important to notice is that we have features from both worlds. Things that we've generated on the MPS platform, they are the crucial ones, and things we've generated with the computer alone. Machine learning for chemistry has done major progress in recent few years. We have very strong embeddings, we call them, but those actually are representations of a molecule, features, properties that are not written in a chemical formula, but are numbers that the machine learning can work with. So we end up with all this data regarding all those drugs that we know their toxicity profile. Then we can train a machine learning model. It's actually an end-to-end deep learning multitask model. We'll go into it a little bit later. But The deep learning is encoding all this data and is able to predict on the training set what will be the toxicity profile. Then, when a new drug arrives, for example, from pharma in preclinical phases, we'll talk about the phases later, we need to scan it on the platform. It's a scan from few days to a few weeks. We extract those features from the MPS. We add on top the computational chemistry features. Then the model that was already trained can predict for this molecule what will be its level of toxicity. Going a little bit deeper about what about those two modalities and what each one contributes, I think the up part is more clear to everyone in this room, the in vitro part, the MPS part, the one that contributes the multivariate matrix of biological markers. So this includes scanning the drugs on our biochip. We have our own proprietary nanosensor that can really measure those signals on those tiny, tiny organs. So as I gave an example for lactate over time, glucose over time, but this list is very large. We have, of course, the imaging modality that is contributing. Imaging has been through years of success in machine learning. This modality is proved to be very strong predictive-wise. The machine learning can see things in images that we as humans will find very difficult to see. So this is the upper part. And we are also considering genomic diversity. We have started doing those scans on multiple single donors to really understand the variance between people doing that. And the in-silico engine on the bottom that contributes another part of very important feature to the system. Those features are a result of the chemical structure of the formula and the machine learning method that's been generated in recent years. This includes a graph neural network embedding on top. This includes those millions of molecules that the world is familiar with and were used so far in the past years to generate those very powerful representations. We also have features that we are computing with both engines. Those are interesting. The pharmacokinetic, for example. Pharmacokinetic is a very important question about drug toxicity. A drug can be toxic, but if the body is very effective in releasing it, in getting rid of the substance, then it will not be toxic to the body. So pharmacokinetic is crucial and a difficult question to predict. So what we do here, we take the chemical structure, we add on top a scan in vitro that expose few features that are related to the pharmacokinetic of the drug. Then we can predict the pharmacokinetic better than using each one by itself. And this is a methodology that we are doing with few type of feature. Pharmacokinetic is one. On top of that, a training mechanism, how the training works. So first there is a step we call pre-training. This is not done versus a specific label, but is done to teach this network something on the vast unlabeled data out there. So each one of the modalities is pre-training on a huge unlabeled data that exists and not labeled for toxicity. For example, the microscopy modality is trained on millions of microscopy images, not labeled to something, but it is teaching the computer what are the most informative features that it needs in order to describe this molecule. So to describe this image, this microscopy image. So image is one example. We've done the same with the chemical modality, training on millions of molecules to both reconstruct the molecule and to predict its properties. You don't need labels for that. You get a powerful embedding even before starting the supervised training. Then we have initial strong representation. We build few deep learning layers on top, and we do supervised learning to predict an aspect of safety. One example for this is drug-induced liver injury. There is an existing data set, known benchmark for the tasks, but this is just one. We are also doing it similarly with mitochondrial toxicity, with BBB penetration, and I will show you later how actually training on all those tasks together is better than looking at each one of them separately. So to start showing you the effectiveness of this approach, the bio-AI approach, generate data, train on the data, predict, I want to start by discussing some of the most known, some of the most important biomarkers for cell viability. So I assume most of the crowd is familiar with ATP quantification as a strong biomarker for cytotoxicity and cell viability. It is reliable, it is stable. The only problem with ATP quantification is that you have to sacrifice the organ in order to have the ATP quantified. So that makes it very difficult to observe long-term effect. You would have to kill many organs in the way of seeing that. It will be very difficult to see subtle changes because you only see what happened in the end of the experiment. For us, the machine learning as the data scientists, it is very difficult to handle this endpoint view. So we have decided that we tackle the fact that we have to sacrifice organs in order to have the ATP quantified. Instead of that, we took the bio-AI approach. And before diving into how it is, I want to give you a first signal for why would it work at all. So on the left side here, you see microscopy image of a liver. It's a 3D liver. This is a 2D image, bright field one, not something too fancy. And on the right side, you can start seeing some of those phenomena that are related to cell death and can be seen from the image. For example, the membrane breakage here, or the color, you will notice that those on the right are darker than those on the left. This is the control. So there is a reason to believe that the image has signals that will tell you something about the cell viability. So based on that, we have decided to use the bio-AI approach and to program the platform to generate a training set. So we have randomized treatment for many thousands of spheroids. We let them live on the platform for a few days. Then we did the endpoint. Everything automatically, robotically. After a few days, we had a training set, few thousands of spheroid images and their respective ATP levels. Then we have trained the machine learning model to predict, given the image only, what is the ATP level of the spheroid. And voila, we have a very strong predictive model. Validated on an unseen experiment. For those of you who are familiar with the paradigm of trained test, this is stronger than that. This evaluation is on an experiment that was never seen, that was not existed on that time. 91% are square errors that are distributed very nicely around zero. Very powerful machine learning model. And this actually changed how we look at viability in general. Instead of having an endpoint view, having this once or twice per experiment, we have it in a near continuous term. Doing bright field image is very easy. So we went from having the view on the left, having, you see on the x-axis concentration, on the y-axis the viability level with respect to the control group, and what we usually had at day 4 and day 7 viability estimation. Now what we have is a continuous viability estimation. We know what's going on there all the time. If we look at the highest concentration, for example, that we could have seen also here that it's dead after 4 days, but it was already half dead after 24 hours. We could have made some decision at that point that will make this experiment smarter. So really this ATP, besides being a better signal for humans to look at, so as I show you here in the graph, what happened and was really interesting and important is that the added features that were created here was very useful to predict another downstream task. So if you think about the large deep learning model, this is a task on the way to predict the viability level, but eventually I want to know the safety profile, whether it will make drug-induced liver injury. So those features that were generated based on the image were highly fruitful also to generate the daily prediction and the safety-related prediction. So both gave us another very strong features to the model, a lot less organoid. We have stopped killing half of the spheroids in the company because of these tools. We have much better views for the biologists, but most importantly, we have much stronger predictive model. The ATP quantification is just one example. The paradigm I told you with the bio-AI, it could work just the same with many other biomarkers. You can think about biomarkers that are expensive, endpoint, hard to do. Maybe we can do it with image. And we are currently in process at Curis of doing many more biomarkers with even more sophisticated microscopy imaging. I think going forward, this will become a very strong, the image-based biomarkers will replace a large portion of the biological ones. We didn't stop here because it's not like I'm coming today to a biologist and telling him, listen, I can do the ATP quantification from image. you can stop killing spheroids. He wouldn't do it. So I needed to convince him. I needed to convince the biologists, the pathologists, and I needed to convince myself that this model is actually seeing what it is supposed to see, that it is doing what it's supposed to do. We wanted to understand that those features that it's relating on are ones that will talk in a near pathological term. So what we did is we trained what we call an explainability layer. The explainability layer goal is to highlight those areas in the image that were mostly responsible for the decision, like the image on the right here. And I know it's a little bit confusing. It looks a little bit like a biological staining. It's not a biological staining. This image was generated completely by the computer. It was never existed before. We call it at Curis an AI staining because it attempts to explain this biological phenomena by using the AI. And again, highlighting the pixels that are mostly responsible for the level of viability. Let's see how it looks throughout an experiment. So what we see on the top row here is a spheroid through 7 days of experiment. On the first day, the spheroid was treated with high concentration of a toxic drug, so we did expect the degradation in viability. On the bottom row, you see the viability level. It is normalized to a control group. It's validated to be highly accurate. By the way, between 0 and 1, because it's normalized to a control group. And what's interesting in this slide is the row in the middle, the explanation that are trying to highlight those areas that contributed the most to the viability level. Initially, in the first few days of the experiment, we see that the explanation layer will mostly highlight those areas that are related to the spheroid morphology, whether how well a spheroid is, its color, how complete its membrane is. You see almost identical to the geometry of the spheroid above. Going through the experiment and using the explanation, we see that it starts highlighting areas that are not even attached to the spheroid. I was struck when I first seen it. I was even a little worried that I have some bug in the model or something like that. But after talking with biologists and pathologists, it was much clearer. This is a known biological phenomena. What the model is able to see here is a debris agglomeration, and not only able to see those debris that went out of the spheroids, it is also able to correlate them. How is it doing ATP quantification? It doesn't know what ATP is. But it is able to correlate those features, the morphological, the agglomeration of debris, to really correlate and understand the level of viability and to give us a very strong model. Okay, so moving on, I want to show you a little few more examples on where our biggest challenges are and how we are using the bio-AI or machine learning on top of those data modalities to solve them. So the first one, translation, maybe the most known one. How do we take in vitro data and in silico data and map it to what will happen in humans? Explanation, I just showed you one example. A high-profile question requires reasoning. It's not a matter of going to the machine learning guy and, you know, this is toxic to 80%. We really want to understand what's going on in there to increase adoption and to be able to move with this forward. The dose response, a difficult task. Most of the data set out there today are labeling drugs for being toxic or not relating to their concentrations, assuming something about the therapeutic range, but what we really want to understand when screening a new drug is the causal relation between the dose and the blood concentration and the toxicity. Any drug can be toxic or not. And generalization, the most difficult part for me. I think 1 or 2 hours every night thinking about that. How can we know that based on everything that we've seen so far, all the data, all the drugs, all the features, is it enough to give an answer about a new molecule candidate? Does it differ too much from what we've seen so far? So we need to be careful with it. Very important question that should provide the boundaries of where a machine learning model is applicable. Starting with the translation problem and going back to a similar methodology as I show you on the first slide, the bio-AI approach, we generate on the left side the in vitro feature resulting from interacting between the drug and the organ, multivariate time series of biomarkers, ATP, lactate, glucose, etc. The in-silico part on the right side contributing some part of the features resulting from the chemical formula. I also put it here, the explanation, although you can say it's partial in vitro and partial in silico. And there are those features that are done jointly by the in silico and the in vitro, for example, the ATP we've just discussed, CYP inhibition we will discuss in a bit, and others. We are pre-training it, as I previously told you, each modality with its own data. And then the supervised learning is to predict the drug-induced liver injury of this molecule. So we are doing the supervised training. We have a training data of about 1,500 molecules that are training the system to predict the diliness level. And of course, we have also the explanations on top. So this is the classical in vitro, in silico to in vivo problem. We have the in vitro data, the in silico data, and the DILI labels that we got from the FDA are post-marketing.Ose are things that were shown on humans to have DILI or not. So this is exactly the machine learning trying to map the signals that we have in the lab and in the computer to what will happen in human. I can show you that on a data set that was published by AstraZeneca in 2017, 110 drugs. We are state-of-the-art performance, better in sensitivity and specificity than any solution before. Now, there are many benchmark data sets out there, so I wouldn't think too much about the numbers themselves. Each data will have different numbers because it is difficult or not, depends on the molecule that were chosen. So when you are told about performance regarding, for example, drug-induced liver injury, the first question should be, on which data set did you test? And this one is tested on many data sets, and I can tell is at least comparable to other on all data sets. I gave one example here because it's a known benchmark and quite comprehensive one. Going to the next phase, I want to talk a little bit more about explanations. And we've previously discussed how Curis is trying to explain, for example, the viability level on top of image, but this is only part of the story. And I want to show you another example here of how we are doing similar thing, but on the chemical structure of the molecule. So previously we said we are predicting DILI. Now we want to explain why is this drug DILI or not. So what we are doing here on the right is we are highlighting those substructures that were mostly responsible for the decision of the DILI. You can think about it as a kind of method to identify tox alert. So we can show that this method is able, without knowing anything about no tox alert, to identify known tox alert. This is aniline, for example, a highly known one. But this actually is an AI method to really find new tox alert and to connect them. What's very powerful about this, so I show you an example of the image explanation, now the chemical explanation. We are training everything on a one deep learning model, which means we are explaining the prediction on top of a joint representation on the image, on the chemistry, on the biology. So think about mitochondrial toxicity, for example. You will be able to see the chemical structure that is responsible together with the damage on the spheroid. So it's the image, the chemical structure, and you'll have importance also of the biomarkers. So we are not just providing a safety prediction. It's not a prediction. It's an explanation with the number, and it should help you to really receive decision. What we are pushing for is to understand the mechanisms of toxicity, because this is the only way that machine learning will be used in large scale. Another very important mechanism that keeps us on ground in the sense of being able to explain and to understand what's going on, we call it the bio-AI feedback loop. So this term encapsulates few very important methodologies in Curis. It meant to say that it is not an AI-based decision. It is something that is done always, the AI and the biology talking together. And few examples for that. The data quality, super important. You can scan on the MPS all day, but do you test the data? We know that biology is noisy. Every data point that's going into our database is first tested to be anomaly or not. What is anomaly? Broad decision, but If it differs significantly from the distribution that we've seen so far, it's not going to the database until a professional is looking at the data, the biologist, he's confirming that what's happened should have, or he's repeating the experiments. No data point is going to the database without validation. Molecular space-driven screening preferences, what does it mean? We are not screening drugs serially. We are screening the model. Every iteration that this model trains, a part of its output is those areas in chemistry that it is less capable of. So we are testing the model on different scaffolds of chemistry to really understand those that it is less capable of. Then we will push them further in the scanning order. We want to cover as much of the chemical space as possible with less example. Eventually we will scan all. But to be able to answer as many questions as we can at a specific time, we are scanning with a smart order. A dynamic study plan, very important component. I will give an example. CYP inhibition, a very important features to identify DILI toxicity. There are many of them. It's very difficult to measure all of them per all drugs. So what do? We give an in-silico score, meaning we are predicting the expected CYP inhibition for this specific drug. We usually can do it, there is no magic number, but for those one we can do it to 70, 80%. Then we are doing a sensitivity analysis on top. Will this CYP inhibition number can change the decision of the model from saying this molecule is toxic to not? We are only interested in getting higher confidence for the important features that can change the decision. If this feature is one that can lean the decision in one direction or another, then it will go to the in vitro scan and the biomarker will be measured in the biology lab. Think about those thousands of biomarkers out there. It makes sense to make the study plan per molecule. Some of those biomarkers, I don't know, a drug that goes to the liver and a biomarker for the brain. Doesn't make much sense. Yes, so I'll conclude maybe just one more word about generalization. Very important aspect of machine learning at all. The most difficult question you should ask every machine learning practitioner, will it work on a new example? I want to briefly say that we are taking a multitask approach here. So if you look at any specific data set, you will find labeled for toxicity, you will find 1,000 examples, 2,000 examples. We have created a cohort of safety portfolio labels based on many, many data sets out there, resulting in 350,000 molecules. And we were able to show that by doing training on many of those tasks, we are predicting better each one of them by training on all of them together than by training separately. And we have increased the chemical space. Going to the end, just 1 minute in a nutshell, Curious in a nutshell. We were founded in 2020, dually based in Boston and Tel Aviv. A strong public collaboration with Merck and AstraZeneca, also with the New York Stem Cell Foundation. Greatly backed by our patents. About 50 people strong, have computer scientists, machine learning scientists, the other have biology scientists. Our bio-AI approach can work in many different phases of drug development, from preclinical early phase where the in-silico part will do the heavy lifting and the in vitro will contribute just small amount of the features, to the pre-IND where the in vitro scan will take over stronger and will provide more features, to the last phase where we are doing the most comprehensive scan in order to do, for example, patient stratification. And with that, I tried to find an image of everybody, but I couldn't, so I bring two. Thank you, everyone. I'm also here for the rest of the day. We'll be happy to talk with you guys. And Amir Bain, our VP Biology, will also be here tomorrow. So thank you very much.
Thanks a lot, Shahar, for sharing the sound of safety. Quite fascinating. Maybe one question?
Okay, quickly. All right, you win.
Wonderful talk. It looks like a beginning of a revolution. So I was wondering like how translational this is, which means that one, if I have one model, one cell line, or one cancer type, could be transferred to other types. And two, if I change, let's say you train a model with your setup, your microscope, if I change to a different brand or slightly change of the setup, will it still work?
Yeah, I will start with the second one. So the work that I showed you about ATP quantification in images will be published soon. So everyone will be able to see it and really understand it better. What I didn't show here is that we have done a major step on top that will generalize this component. This can work for every lab, every microscope. I will not get into the machine learning because this is a big part of the contribution there, but this work will be published, and with this model and trained parameter, they will be on GitHub. You can take it to your lab and it will work. And regarding transferring between diseases, between tissues, major question related to generalization. I would be careful, as I've seen machine learning in its top and in its bottom. I would be careful. You need to scan smartly. And I would say that when you ask this question, you're already on the way to the solution because you understand that there might be a problem.